Hello everybody, I'm Devon.
I'm John.
And our project is centered around using nanobots to treat anxiety.
And our mentors was Professor Ukar and Dr. Green.
Alright. So, quick introduction to opioids and what they are. Opioids are chemically related drugs that interact with the opioid receptors on nerve cells in order to trigger specific emotional responses. There are three main types that are very specific to the central nervous system, which are the alpha, delta, and mu receptors. The opioids. Opioids are generally safe as long as you're taking them as a prescribed moderated amounts, because there are very mild opioids, but there is a very common misuse of them, such as opium back in China. Most of us are aware of that from history and the Opium Wars. And that causes a very serious issue. Knowledge about the chemical exchanges between opioids and cells help scientists figure out interactions of the neural type and the overall function of the brain just generally, because it's a key function. And opioids help patients dealing with chronic diseases and disorders because opioids are generally used for pain treatment.
So our first objective was to understand how opioids affect us on the nanoscale. If we're going to implement nanobots on the nanoscale, we first have to understand how opioids are reacting and being traveled through our nervous system. So the first thing that we decided to do was research which opioids were exogenous and endogenous to humans, found in plants, found in our bodies. If we naturally produce them in our bodies, we can naturally implement them. We can actually induce them through drugs and implement them through our nervous system. So the whole point of implementing the nanobots into the opioids was to monitor and see cells conditions and cell development, and as well as control chemical releases throughout the body. And our hypothesis was nanobots can carry opioids through the bloodstream to target and latch on to delta opioid receptors specifically. This can be shown using a conceptual framework.
So the steps we will be taking. The first one will be to determine which anxiety-related opioids receptors need to target, which in this specific case would be the delta opioid receptor. From there, we need to figure out how to implement it. And we learned that gold nanorods can be attached to strong opioid drugs, such as deltorphin, which only target cells with delta opioid receptors. And the research concluded that pretty much all of the gold was redistributed to a vast amount of the cells that were expressing this delta opioid receptor. So if we modified this experiment to carry opioids along with nanobots to specific cells which are exhibiting the symptoms related to anxiety or stress, then we can easily influence these hormones that are being transferred from neuron to neuron, as well as record the data of what is actually occurring on the nanoscale.
So nanobots have a huge impact to construction and implementation of cells in the human body. We basically can monitor and redistribute certain chemicals in our body that make us react to certain stimulant. And so, if properly attached to individual cells, nanobots have multiple applications, and more research is needed, but this application can further knowledge about translation and transcription.
And this is a list of our sources that we used.
And we plan on 3D printing a nanite that would specifically attach onto the gold that was distributed to these specific cells. And it would be obviously a larger scale, it wouldn't be, a nanoscale nanite, because then nobody would be able to see it. So, yeah.
Any questions or comments?
Yes, well,